DNA is the software of life. These organisms that we're finding in the ocean are DNA-driven biological machines. You take away the DNA and the cells die. You change the DNA, the cells change into something else. It was a mapping expedition, but he was just a biologist on board, but he was just asking questions about diversity of what he could see. So the difference is we're looking at the world that Darwin couldn't see. You know, if you look at that water, it looks pretty clear, right? You wouldn't assume that there's about 100 million species right there, right next to the boat. You say, oh, it's pretty clear water. But that's what we're doing. We're cataloging information now using the genetic code as the means instead of taking a picture or drawing pictures. He didn't take pictures, he drew pictures of all the different species that he found. He collected them. So if we found birds, you know, he killed them and, you know, saved them. So now it's very different. We don't need the species, we don't need pictures of them, we don't do drawings of them. We have their genetic code. As you said, it was 10 years ago that we announced the first draft. It took 7 years to, so 2007 we published the first complete version of the human genome, so that's much more recent history. That's only 3 years ago. And we're learning what we expected, that the human genome is much more complicated than most people thought it would be. You know, people early on, 10 years ago, were looking for simplistic explanations that there was one gene for every trait. And that's why people expected hundreds of thousands of genes. So the first big surprise is we only have 20,000 something genes. Right? We don't know what most of those genes do yet. We don't know how they work together. So we're very early in our knowledge. We're working on how to, what's called haplotype phasing, so you can separate your parent chromosomes from each other, so you could actually know what you inherited from your mother versus your father. Working at the most complicated stage of how to get all the phenotype information. The sequencing the DNA is now the easy part. Correlating it with everything else in your life is very complicated, so we're trying to start on the early parts of the next phase. Well, what I'd like to see is obviously all the things we've developed, like the first human genome, the first synthetic cell, understanding all this life on the planet, to lead to some real solutions. Solutions for healthcare, solutions for decreasing CO2, maybe new food and fuel. It's the beginning potentially of a new industrial revolution, a scientific revolution. That would be nice to see the results of that.